Good afternoon. It's my pleasure to present this talk to you titled Meeting the Challenges of Randomised Multi-Centre Clinical Trials, Experience from PSMA Imaging and Theranostics in Australia. My name is Michael Hofman. I'm a nuclear medicine physician from the Peter MacCallum Cancer Centre in Melbourne, Australia. I'd very much like to be there in person with you at the NCI. But this was not possible, so I hope you enjoy this video version of my talk. These are my disclosures. I did the first PSMA PET-CT at Peter Mac in our centre back in 2014. Here's a patient. We can see the primary tumour really clearly seen on the PSMA PET, along with a nodal and distant metastases. And after you do maybe 10 or 20 of these scans, you become fairly convinced in your own mind that this is superior to standard imaging. But these sorts of case examples are certainly not enough to convince our regulatory authorities or funding authorities that we should switch from conventional imaging to PSMA PET. So it's emerged that PSMA PET is potentially a new gold standard. It may supersede other imaging as a one-stop shop. At least in Australia, it's widely potential for wide availability at low cost. We get images with extraordinary high tumour to background contrast, and despite thousands of these scans being done in Europe and Australia, there's very little prospective data on accuracy or improvement in patient outcomes. So we have conducted the PRO-PSMA trial, a prospective randomised multi-centre study to evaluate the impact of gallium PSMA PET-CT for staging high-risk prostate cancer prior to curative intent surgery or radiotherapy. This study has run at 10 centres around Australia. A lot of investigators, and I won't go through, but I will tell you that it was a close collaboration of nuclear medicine with the surgical oncology teams that were referring these patients, also radiation oncology and also medical oncology. And this was one of the keys to the success of this study. So in this trial design, we take patients with high-risk prostate cancer. They are screened and then randomised to either PSMA PET-CT or standard of care, which is a bone scan with SPECT-CT and a contrast-enhanced CT. And then patients cross over to the second-line diagnostic imaging, consisting of the alternate arm, unless there are more than 3 distant metastases, in which case there's not much point crossing over. And then we use follow-up up to 6 months, including repeat imaging, to establish the ground truth. And in a select group of patients who have negative PSMA PET scans, we're continuing ongoing follow-up to evaluate the prognostic value. This study started in 2017 and recruited quite well. In fact, in the last quarter of the study, well ahead of schedule, and the randomization was completed around 6 months ahead of schedule. I'm pleased to tell you that the results of this have just been analysed, and we've just submitted this to a conference and hopefully present the results to you in the first quarter of next year, hopefully with a publication as well. How did we get there? How did we conduct a 10-centre, 300-patient randomised imaging study? Well, I would say collaborative studies are a team sport. A lot of things need to come together. This is not a one-man exercise. We need investigators, and we need not only nuclear medicine physicians, but we need our radiochemists, pharmacists, technologists, physicists, and we need to work very closely with our clinical specialists, particularly in assisting with study design. We need to get the institutions together, and another critical component are clinical trial groups, and I'll go into these in a little bit more detail. Industry collaborations. We must have a coordinating centre. This is not a single person exercise to run a study at 10 sites. We need a dedicated study coordinator, a biostatistician. We need to know some of the rules about conducting clinical trials, a rule book called GCP, or Good Clinical Practice. We need a database set up, electronic nowadays, and we need these individuals to be independent without any conflicts of interests. And obviously we need a grant, and there are various sources from government to philanthropic, industry and institutional. And most importantly, I think we need a principal investigator who's like the team leader to bring all these people together and drive everything, make sure that the timelines are met, keep everyone on. One of the critical elements to this study was ARTNet, the Australian Radiopharmaceuticals Trials Network. This is a member of the ACTA, the Australian Clinical Trials Alliance. This is a collaborative network incorporating medical specialists, technologists, scientists, researchers with a shared interest in multi-centre clinical trials that utilise radiopharmaceutical for imaging or therapy. The mission statement of ARTNet is to promote, facilitate innovative, collaborative clinical research. I'm one of the scientific members of this organisation, and they've been pivotal. So what have the ARTNet done? I think quality assurance is critical in multi-centre trials. So in this ProPSMA trial, prior to commencement, all sites underwent PET camera phantom validation. Since we were using gallium-68, this was done with gallium-68. And a lot of sites had validated PET scanners for fluorine-18, for FDG. And in the beginning, I thought, is this really a useful exercise to send phantoms around sites, to fill them with gallium and measure things? And as we'll come to see, it was extremely useful. We also had to set some standardized radiotracer production parameters for gallium PSMA, because these were produced in hospital radiopharmacies. And then we had some quality control during the study that we'll go into. So gallium-68 PSMA validation testing, through ARTNet we got a group of physicians, chemists and pharmacists together, and we came up with the minimum specifications for production that all sites could meet. This did incorporate some fairly high-level QC, so HPLC in all patients. Sites had to perform a minimum of 3 consecutive successful test runs with full quality control and get a certificate before enrolling patients on the study. And then during the conduct of the study, for each production run, the batch release parameters were a little bit less arduous, but still incorporating HPLC. And all this is logged and carefully recorded. The other thing I alluded to was we did PET-CT validation, where a standard NEMA phantom was sent around to sites and filled with gallium-68. These were analysed by a imaging laboratory, and unexpectedly, what we found was that the dose calibrators around Australia, and for that matter of fact around the world, were not accurately measuring gallium-68. So the amount of gallium-68 that was being administered to patients on the basis of the standard dose calibrator settings. This publication in JNM to find out a bit more, but actually 10 of the 14 PET centres underestimated the SUV by 15%. This was the Kapanek dose calibrator, and we adjusted the default parameter and corrected for this, but it highlighted how testing the entire chain of measurements and calibration is pretty critical in these multi-centre trials and should form part of the pre-qualifying site assessments. In Australia, we don't have the FDA, we have the TGA, the Therapeutic Goods Administration, and we are fortunate to have a system called the Clinical Trials Notification, or CTN scheme. This is the equivalent of your FDA IND scheme, but it's very different. This is a notification process. So unlike your FDA IND, where you submit an application for a new compound to do a clinical trial, and the FDA assesses that, here we really just notify our government that we are doing it, that the intent is to use an unimproved therapeutic good, and they rely on the local Human Research Ethics Committee, our HREX, to review the scientific validity. The TGA do not evaluate this, and the institution becomes the approving authority and gives the final conduct to undertake the clinical trial. And this has enabled us in Australia to conduct many of these novel clinical trials using new radiopharmaceuticals for imaging or therapy. And if you have some FDA people in the audience, my message to them would be that you should look at the Australian system, particularly in the nuclear medicine sphere, because it does enable these clinical trials to be performed with a lower level of oversight, but what I would purport is an appropriate level of oversight, since the risk, particularly from a diagnostic radiotracer, is really very low. In our study, all the PSMA PET-CTs were also centrally collected and underwent central review. These were our secondary objectives, and I won't go through them, but they included management impact, safety, equivocal study rates. We've got some health economics, reporter variability, radiation dose to patients. But I want to focus on the primary objective in this study, which was to compare the accuracy of PSMA PET to conventional imaging. This is not so easy to define. So in this patient with widespread bony metastases on PSMA PET, if this patient underwent a bone biopsy and the bone biopsy came back no malignancy, would you believe the result? And if you did, and if you set histopathology as the reference standard, this would be classified in the study as a PSMA PET false positive, which clearly it is not. This would actually be sampling error. So we thought about this long and hard, and we came up with a truth panel for the gold standard, consisting of 1 hard criteria or 3 soft criteria. And I'm not going to go into this in any detail, but our clinical trial protocol has been published in the British Journal of Urology. So please feel free to look this up and use elements in your next clinic. So I'm going to change tracks now to therapy, to theranostics. And this is something I've had an interest in for a long period of time. Extensive experience in our centre with lutetium dotatate for treating neuroendocrine tumours, a program set up by Professor Hicks. This gave us the infrastructure to do gallium imaging and lutetium therapy. And when PSMA came along instead of dotatate, it was fairly easy for us to switch across to a new compound. What I had learned in my experience in neuroendocrine tumours since really my time at Peter Mac was that despite treating hundreds of patients and publishing results of these, oncologists do not believe retrospective data. So I made a conscious decision that all our PSMA therapies would be done as part of a prospective clinical trial. So this was not a multi-centre trial, but we did a very simple 30-patient prospective trial, and this was published in 2018 in Lancet Oncology, and we really showed remarkable responses in patients who progressed after conventional therapies. But the message here is that if I had done a several hundred patient study, retrospective, without inclusion criteria, without exclusion criteria, it would not get into an oncology journal. It would end up in a nuclear medicine journal. But if you conduct a well-designed study, even with a small cohort, you have a good chance of getting into a highest impact. Now, we showed response rates, PSA dropping over 50% in 64% of patients. The results of this, the updated results of this, were published in JNM online just a few days ago, so please look that one up. And it produced this beautiful image of the air, PSMA PET scans before and after lutetium PSMA therapy. This is in the 8 patients with the best responses, and received the S&M image of the year in 2018. Interestingly, this image was included in the Lancet publication, and at an editorial level they asked us to remove the image. This was because they felt we were cherry-picking the best responders, and it highlights how our oncology community is not really influenced by a few impressive images, but they want some hard responder in this patient. Really spectacular images with PSA dropping to undetectable, but oncologists will not believe anecdotes and they will not believe retrospective data, but they will believe Kaplan-Meier survival curves. This is what they want. And we showed an overall survival of 13.3 months, and this was great data. It led to the Lancet publication, but it's certainly not good enough to make this therapy reimbursed or even registered. For that, we need a randomized trial showing that this improvement in overall survival is better than an existing standard of care. But the Lancet publication, the track, enabled us to apply for a grant through ANZUP, the Australian New Zealand Urology Clinical Trials Group, and PCFA, the Prostate Cancer Foundation of Australia, leading on from the PRO-PSMA trial, to use that established network to run the therapy trial, a randomized phase 2 trial of lutetium PSMA 617 compared to carbazitaxel in patients with progressive metastatic castration. This trial is using that established network, so it was so important to get that first trial up and running, we could take a lot of the elements of the pro-PSMA study. So for example, the PET scanner validation, the PSMA, the gallium PSMA QC, we just rolled that straight into this. In the study design, patients have a PSMA and an FDG PET, and if they're suitable, they're randomized to lutetium PSMA or carbazitaxel. This clinical trial protocol has also recently been published online in the BJUI, so you can look that up if you're interested in the details. Again, we used ArtNet to certify sites prior to production. This consisted of QC for gallium PSMA 11, but also a newly developed QC for lutetium PSMA 617, which was produced in our hospital radiopharmacy, and again, PET scan validation. For this study, we produced a therapy nuclear medicine manual, which went through elements that all sites had to comply with around PSMA PET, lutetium production, central review and image storage, and radiation precautions. Similar to the QC for gallium PSMA, we used the same team to develop standardized QC for lutetium PSMA 617, again with all patients undergoing HPLC quality control. As I alluded to, all patients underwent a PSMA and FDG. We pre-specified the inclusion criteria. So for example, patients must have had an SUV max of 20 at a site of disease on PSMA, and not all patients were suitable. Here was a patient with low PSMA expression or a patient with this mediastinal mass with no PSMA uptake but high FDG uptake. In this trial, we used the Widen system to centrally collect all the imaging data. This is a great system that we've used for this trial. Sites upload their scans online. The system automatically anonymizes the study, does a quality control check, and it then sends an email out to the central review committee to say an image is ready to review, and we do that blinded to start with, but if there's no consensus, we get a third reviewer. We have a discussion about it and unblind it, and then we end up with a result that's sent back to the site to let the coordinating centre know if the patient is eligible or ineligible. Now, this study has also run very well. This is a 200-patient study, and we randomised the 200th patient at 10 sites around Australia at the end of August, and this also ran 5 months ahead of schedule, and we're now in the follow-up period, and we hope to perform the first analysis of this study early in 2020 and maybe present the results in the middle of next year. Our focus has now shifted from moving lutetium PSMA from a last line of therapy to an early line of therapy, and now that the therapy trial has completed patient randomization, we've been looking towards the next trial, and from a DoD grant together with a grant from Movember Australia, we've put together the upfront PSMA trial. This will be a trial, first trial of lutetium PSMA in patients with hormone-sensitive disease. We'll take patients with newly diagnosed metastatic disease and randomise them to standard of care, ADT or docetaxel, or the therapeutic intervention of lutetium PSMA plus ADT. We hope to commence this study in January. So this is my second last slide, and it's just highlighting a key element of doing these multi-centre studies is engaging with the multidisciplinary team. And one of the things we've accomplished at Peter Mac is that we work very closely with our urology, radiation oncology, medical oncology colleagues, and that's really the only way to do these multi-centre trials and get them going. So there's a lot of people behind this work that I'd like to thank. People at Peter Mac, our uro-oncology multidisciplinary team, our funding partners, a particular shout out to Movember, Prostate Cancer Foundation for supporting this work, and our industry partners, and our collaborative research groups that we work so closely with. Picture of the uro-oncology team, our nuclear medicine team, and our prostate laboratory team. Thank you very much for listening. If you have any questions for this talk, feel free to send me an email. Thank you.